Let's take a look at the structure of DNA. First of all, DNA stands for deoxyribonucleic acid. Deoxy is the sugar, nucleic is the nitrogenous base or the nucleotide, and acid is the phosphate. It is a polymer. It is made up of repeating nucleotides, which are the monomer. The nucleotide is made up of three different parts. The first part is the nitrogenous base, and these are adenine that pairs with thymine, guanine that pairs with cytosine. This is called the base pair rule or Chargaff's rule. Another way to remember that is A pairs with T because they're sort of angular and pointy letters, and G pairs with C because they're curvy letters. That's another way of remembering them. The second part of this is purines is a term that we collectively call nitrogen bases with two carbon rings, and these are guanine and adenine. My learning tool for this is that I think of water as pure. Agua is Spanish for water. It is also pure. So A and G both have two carbon rings, whereas pyridamines have one carbon ring. And likewise, this is cytosine and thymine. So across in a pair of DNA, you're going to see, or one base pair is going to have a total of 3 carbon rings. The second part of a nucleotide is the deoxyribose sugar, which is found in DNA. And the last part of a nucleotide is the phosphate. And again, the phosphate is what makes DNA an acid. Here is a model of DNA that students built. So you can see that the rungs of the ladder, so here's guanine and cytosine, adenine and thymine. You can see that these are longer, so these are the ones that have the two carbon rings in them. You can see that they connect to a deoxyribose sugar, and that in between each of the sugars is a little phosphate that connects them. The nucleotide, this part of the nucleotide is held together with covalent bonds. However, the bond in the middle is held together by a hydrogen bond. Again, DNA is a long strand that comes apart in places so that the genetic code can be read. So DNA is read from the 3' to the 5' end, and this has to do with the way the sugar is pointing. So example, DNA is read from 3' to 5'. So I think of it as 3', it's getting bigger, so I read it in this direction. However, DNA is built from 5' to 3' because these two sides are always opposite. I like to think of DNA as a ladder, and I use this analogy. The sides or the backbone or the that you would hold on to, the vertical pieces, are basically the phosphate and the sugars, and the rungs that you would hold on and climb up with are the pyrimidine bond with the purine bond. Again, these bond in the middle with hydrogen bonds because they're a weaker bond, whereas the bonds on the sides or of the ladder are covalent bonds. Another thing that you should be aware of is that for every 3 letters or nucleotides, every 3 of those is called a codon, meaning it codes for a specific amino acid, and there are 20 amino acids. And again, thinking of amino acids, together amino acids are monomers of proteins. So like the nucleotide builds DNA, amino acids build proteins. The last part of DNA is the advantage of DNA. It's a fairly stable 3-dimensional shape because it has what's called a double helix. It actually twists on itself. If there's a mutation or a problem on one side of the DNA, there is a backup copy, and DNA is constantly double checking. And also, DNA is a larger molecule, and so it can never leave the nucleus. It is so precious, we lock it in the nucleus. Another thing about DNA that people are not aware about is that your DNA is identical from cell to cell in your body. It is called your genome, also known as the blueprint of life. Cells in your eyes, your heart, and your skin have all identical DNA. However, only certain portions of that DNA are being read and used at that time. The only different DNA in your body is DNA that is found in sperm and eggs.